Molecular mimicry is a complex challenge. It is when different proteins share a common domain, and this can confuse the immune system and cause it to react against both of the proteins, even though only one of them is foreign, leading to breakdown of tolerance and autoimmunity. I wonder if computational tools can help us with this.
Well, if you have the two proteins and their sequences, then it's easy. This is what we learned last unit with sequence alignment. I guess you are referring to this situation where you suspect biomimicry, but you have only one protein, and you can't find the other protein that is similar to it. In order to explain this solution, one needs to understand the scientific context. The big picture here is organizing the world's biomedical information. Your need to identify the second protein requires to search that sequence against a database of all the proteins known in the world.
Yes, so we spoke about classic sequence alignment last week. It's quite fast due to dynamic programming, but it will still take an unreasonable amount of time to do a comparison to the millions of known proteins.
Let me explain with an analogy from the digital revolution. Google's search engine. You can search for a very specific term across the enormous database that represents the internet and do that in a split second. This is thanks to a methodology called pre-processing, which is done to every record before it can be searchable. It uses advanced mathematics to compute a tiny fingerprint that represents the entire record. The database is actually organized according to these fingerprints, what enables a very rough initial filtering just by comparing fingerprints, which is a very fast process, better in several orders of magnitude than sequence alignment.
I guess you're hinting at BLAST, the most well-known bioinformatic tool. So I understand that the underlying technology is similar to Google?
The mode of action is similar, but they are also similar in another aspect, the impact they made to the world. We could have not done what we do now in medicine without these biological databases and the BLAST search algorithm.
That's right.
And this is not where it ends, it's only where it starts. Once finds a protein, you can use genome browsers. These are great tools that enable you to see all the accumulated information in the world about a specific protein in the context of its genomic location. And this is done by multiple layers of information.
Genome browsers are very important because they incorporate clinical databases that describe human variation and link it to human disease.
Ron Unger.
Again?
He can give a fantastic explanation about biological databases, how to search them, and how to display the results.
He will. Okay. And I will see you next week in another Spotlight session.